What's up, biochemists? Today's lecture is all about fatty acid catabolism, how we break down and digest dietary lipids and then burn them for energy. If you enjoyed the citric acid cycle lectures, I think you'll have a fun time here because there is considerable overlap between these two forms of metabolism. We will be first describing how the mammalian body absorbs and distributes fats from the alimentary canal to the cells in our bodies. After that, we're going to illustrate how fatty acids are catabolized and shunted into oxidative pathways. We call this process beta-oxidation. And then to end us off, we will be describing how the products of beta-oxidation are then delivered throughout the body. Today's molecule of the moment is actually the mystery itself. I want you to identify it based off of the following clue. Now, you may have noticed that most saturated fatty acids that we have mentioned in class so far, lauric acid has received a lot of attention. It's a C12. Myristic acid, C14. Palmitic acid, C16. You'll see more of that next time. And stearic acid, C18. They all feature even-numbered hydrocarbon chains. I want you to review the chief metabolic intermediates featured in class so far and propose a simple explanation, such as a common building block between all these compounds. And maybe once you do that, you'll be able to infer why fatty acids are constructed or broken down in 2-carbon intervals. Fatty acid catabolism, as you gleaned, is all about converting fats into energy, ATP. And it's a common misconception that you need to be in a prolonged caloric deficit in order to burn fat. And that is not true. You need to eat fewer calories than you take in order to lose weight. But you burn fatty acids every day. In fact, 40% of your daily ATP expenditures come from fatty acids. And in the heart and liver, that figure is doubled. 80% of the energy that fuels mammalian heart and liver tissue comes from lipid sources. Fats are crucial to our well-being. So where do we get them? The three major sources are, of course, going to be our diet. And when we consume lipids in our diet, they end up getting stored in adipose tissue. This is a picture of white adipose tissue. And then, of course, anabolism. We can make lipids from other biomolecules. If I feed a rat a large caloric excess of glucose and only glucose, it will gain body fat over time because it will convert and store the excess energy as lipids, even if it wasn't fed any. Internalizing lipids does pose a minor conundrum to our bodies. We know that fats tend to be rather hydrophobic, and we ourselves are aqueous organisms. So, how do we absorb and shuttle such hydrophobic entities throughout our hydrophilic cells? It's a multi-step process, but the first step basically begins with mechanical digestion, and that occurs in the small intestine. What that entails is the gallbladder will inject these bile salts into our duodenum, and these are basically charged small biological soaps that are going to help bust apart the fat globules in our chyme. What you may see in this graphic over here is that these biomolecules are going to coat the fat globules with a negative charge, shown as these green circles. And the acquisition of negative charge around these smaller fat droplets will prompt them to shrink into even smaller micelles, which helps space apart the negative charge a little bit better. We call this process emulsification. And again, let's repeat. What does that entail? The bile salts are going to increase surface charge. That is going to cause the micelle size to decrease. And when you shrink the micelles and you get several tiny ones, we are actually increasing the surface area. And that's the key to step one. That surface area increase is really important because it's going to allow for step two, intestinal lipases, to chemically digest the triglycerides within these fat droplets into glycerol and fatty acids. The third step of fat trafficking occurs within the mucosa, or the lining of the small intestine, and cells called enterocytes are going to absorb these fatty acids that have just been released into the lumen. And once we do that, it's actually going to recombine them back into triglycerides. Let's follow along that process in this diagram over here. In the intestinal lumen, we have the bile busting apart our triglyceride globs into triglyceride micelles. Intestinal lipases are going to break that down into free fatty acids and glycerol. The enterocytes, the cells lining the mucosa, are going to take both of those in and reform it back into the triglycerides. So I know what you're thinking. That seems a little bit pointless, but there's actually a really good reason for that. What happens next is the enterocytes will take the triglycerides they just reformed and it's going to package them into these special aggregates called chylomicrons. These are formed when you combine something called apolipoproteins along with cholesterol and phospholipids, and you make basically a giant sack filled with triglycerides. These are going to be much larger than micelles. The reason we emulsify and break down the triglycerides in the gut is so we can absorb them easier, but it's easier to transport fat throughout the body as triglycerides because they're not going to be charged anymore. Fatty acids are going to have a negative charge, and that is going to prevent their micelles from getting very large, thanks to all that negative charge repulsion. Triglycerides, however, are not going to have any charge or as much of a charge. And as a result, we can pack more into a single chylomicron, which means we can get away with making fewer of these chylomicrons per meal, which is going to be much more manageable to ship off and deliver through our lymphatic system. As foreshadowed, the last phase of fat trafficking is going to occur throughout the circulatory system. These chylomicrons that we just formed in our enterocytes are going to be dumped into these collecting ducts called lacteals. And lacteals are basically small lymphatic vessels that will eventually feed into our greater lymphatic circuit. From there on out, these chylomicrons that we just packaged and made are going to be diluted in lymph, and then that lymph will be slowly introduced into our bloodstream through the subclavian vein. And the key word here is indeed slow. we want to trickle in this fat-rich lymph because if we dump it in all at once, it could precipitate as this fat clog, and we certainly don't want that happening inside of our veins. But so long as we're patient, we're going to get the lipids successfully suspended in our bloodstream, where they can circulate to the tissues that need them. And that's where step 6 is going to kick in. When tissues demand fats or fatty acids, they will secrete an enzyme called lipoprotein lipase, and they will secrete that into the surrounding capillaries. What this enzyme does is it will disassemble the chylomicron and redigest the triglycerides back into free fatty acids. So there's a lot of ping pong here between fatty acids to triglyceride and then back to fatty acids again. But again, we can't have fatty acid micelles just forming spontaneously in our capillaries. That's where serum albumin comes in, and I have a picture of it right over here. Albumin has many roles in the bloodstream, and one of them, believe it or not, is to actually help carry fatty acids safely by binding up to 7 of them at a time, shown over here. These fatty acid binding pockets are going to protect the bloodstream from having fatty acids just precipitating within it. And lastly, the final step is where the fatty acids finally enters the cells that need them through special fatty acid channels. And because biology is smart, fatty acid uptake is going to be hormonally regulated. When we are in the fed state and energy is in surplus, insulin will cause lipoprotein lipase secretion to increase from adipocytes. Conversely, it's going to reduce LPL secretion in myocytes, muscle tissue. So what we're doing is directing the flow of fat traffic from the sink tissues to the source tissues. When energy demand spikes through hunger or stress, glucagon and epinephrine respectively will direct those fatty acids away from the source tissues and into the sink tissues. It's going to promote LPL secretion in myocytes. So glucagon less epi is going to cause LPLs to rise near myocytes. And you might be wondering, well, does glucagon and epinephrine also decrease the production of LPLs near the adipocytes? And surprisingly, the answer is no, it does not. There's no significant alteration occurring there. And there's actually an article that was published in 2022, and the title says it all. The title is actually Glucagon Receptor Signaling at white adipose tissue does not regulate lipolysis. 2022. Which means that glucagon doesn't seem to increase or decrease the amount of fatty acids taken up by fat tissue. And this is going to reinforce a common theme that biology isn't perfect, it just needs to be good enough. And this slide is a nice visual summary of the 7-step process of fat trafficking, beginning with bile salt emulsification of the ingested lipids, then the chemical breakdown of those fat micelles into glycerol and fatty acids, uptake of the fatty acids and glycerol into the enterocytes, repackaging of those fatty acids and glycerol back into triglycerides and then into these chylomicrons, transfer of the chylomicrons through the lymphatic system and then eventually the circulatory system through the subclavian vein, site-specific disassembly of the chylomicrons by secreted LPLs, lipoprotein lipases, and then finally, fatty acid migration into the cells that need them. Now that we've described how fat can actually enter respiring cells, the next challenge is getting the fatty acid inside the mitochondria, because that's where we can fully extract its chemical energy through catabolism. The problem is, though, that any fatty acid that's considered long chain, let's say longer than 10 carbon units, isn't able to diffuse freely across the inner mitochondrial membrane. So we need to activate long-chain fatty acids first by conjugating it to coenzyme A within the cytosol. And the enzyme responsible for this transformation is fatty acyl-CoA synthetase. This is a two-step ATP-dependent enzyme that is going to first coax the carboxyl end of the fatty acid to nucleophilically attack an ATP molecule, like so. It's going to attack that phosphorus, and it's going to induce the cleavage of this whole pyrophosphate moiety, giving us a high-energy fatty acyl adenylate intermediate, and this is going to be bound to the enzyme. From this point forward, this high-energy intermediate is going to be susceptible to a second and final nucleophilic attack by coenzyme A itself. Its free thiol is going to attack this electrophilic ester carbon over here, and then this part of what used to have been an ATP molecule is then going to serve as a leaving group, leaving us with a still decently high energy fatty acyl-CoA. And the whole delta G of this reaction is negative 15 kilojoules per mole, and that is due in part to the hydrolysis of the ATP molecule that helped kick this all off. Now, that might be very nice, but you may be wondering, well, what is the point of this fatty acyl-CoA? The point is that coenzyme A is an even better leaving group. We are going to use a second enzyme called carnitine acyltransferase 1 to swap out the CoA we've just installed with something called a carnitine group. And this here is a picture of what carnitine looks like. We do this because the mitochondrial membranes have special channels called acylcarnitine code transporters that recognize carnitine really well. And their job is to specifically pump fatty acylcarnitine into the matrix. And as a follow-up to that, it's going to pump out free carnitine only. Carnitine goes out. Fatty acylcarnitine goes in. And lastly, on the inner mitochondrial membrane, we have a second carnitine acyltransferase, which does the opposite of the first one. It reactivates the fatty acid by putting the coenzyme A back on it, and it recycles the carnitine. So this is the fatty acylcarnitine we just imported. We will take coenzyme A floating within the matrix and do a switcheroo. Bam. We end up with what we began with, fatty acyl-CoA. And then that carnitine is free to be recycled. This whole process is called the carnitine shuttle. And this is incredibly important because this is the rate limiting step. For fatty acid, catabolism. This is a tightly regulated process that's actually inhibited by the substrates involved in fatty acid synthesis, which will prevent the wasteful simultaneous anabolism and catabolism of fatty acids within the cell. In this slide, I have a graphical overview of the pathways that will happen next. Here within the mitochondrial matrix, the cell is now able to catabolize or oxidize the fatty acid for energy. And the first step is called beta-oxidation. Sometimes it's known as the fatty acid spiral. You'll see why soon enough. And basically, it's a series of reactions that are going to chop up the fatty acid 2 carbons at a time into acetyl-CoA units, just like so. And as we do that, naturally, we are going to generate several equivalents of acetyl-CoA. For a C16 fatty acid, we are going to make 8 acetyl-CoAs in total because they are going to contain 2 carbons apiece. Beta oxidation will also generate a fair amount of NADH and FADH2. Don't forget about those. And oh look, when we make acetyl-CoA within the matrix, that can just conveniently slip right into the citric acid cycle, which we call acetyl-CoA oxidation, right? Doesn't that sound familiar? And what's the whole point of the citric acid cycle? It's to chop down and break apart acetyl-CoA into, sure, CO2 molecules, but also NADH and FADH2. And look at that. We can converge these metabolic pathways of glucose metabolism and fatty acid metabolism and converge them onto stage 3, which is NADH and FADH2 oxidation. Doesn't that also sound familiar? This is going to occur within the inner membrane of the mitochondrion. We will take the copious quantities of NADH and FADH2 generated from stages 1 and 2 and dump them into the electron transfer chain, which is going to power oxidative phosphorylation. So it all comes to a head. Okay, fair warning, the next few minutes are going to be a little bit of a doozy, but in my opinion, I think learning about beta oxidation is well worth the effort. We sometimes call the process of beta oxidation the fatty acid spiral because it is going to represent a series of reactions that repeat cyclically, kind of like the Krebs cycle, but unlike the Krebs cycle, they won't go on forever. We are eventually going to spiral out and run out of substrates, and that will become apparent in a few moments. I want to first talk about the goals and the general motions of the fatty acid spiral before we dive into the chemical logic of the 4 enzyme catalyzed steps, which are namely going to consist of an oxidation, a hydration, a second oxidation, and then a cleavage reaction. So what is the goal of the fatty acid spiral? We are going to take our activated fatty acid, which is a fatty acyl CoA, and we are going to digest it down via 2 carbon increments with each repeat of these 4 enzyme catalyzed steps. And the way we do that is by oxidizing the so-called beta position, hence beta oxidation. This over here is the acyl carbon. That is the alpha carbon, which is 1 carbon away from it. And next to that is the beta carbon, 2 carbons away from that. And our goal throughout this entire process, this spiral, is going to be systematically oxidizing this position. And by oxidizing that carbon, we are not only going to get energy out of this molecule, but we are going to weaken it considerably at this highlighted alpha-beta carbon-carbon bond, to the point where it's going to become so weak that we can rip this entire part off and generate this unit as a leaving group, a coenzyme A attached to two carbons. And hey, that kind of sounds like acetyl-CoA to me. So one turn of the fatty acid spiral is going to shorten or digest our fatty acid by one acetyl-CoA unit in the head to tail direction. And this will be repeated iteratively until we run out of fatty acid, essentially. And this cycle is able to be repeated a good number of times because by destabilizing the molecule through oxidation of the beta position, we're essentially swapping out the acetyl-CoA portion I've highlighted in yellow with a brand new coenzyme A that takes the place of it and links onto the leftover beta carbon. And I understand that probably sounds a little bit nebulous at the moment, but it will hopefully make more sense as we dive in. I'm just going to clean up this molecule and then say again that the motions we're undertaking are going to consist of first an oxidation. We're going to oxidize this bond into a double bond. Then we will hydrate that double bond into a beta alcohol. Then we will oxidize that alcohol into a ketone, which is going to make this very unstable and easy to break apart and generate an acetyl-CoA fragment and a fatty acyl-CoA again that has been shortened by two carbons. So let's begin and now talk about the chemical logic now that we have talked about things from the bird eye's view. I'm going to clean this up again. I'm trying to be deliberate here with my color coding to help you follow along to the best of our abilities. There. And let's start. The first oxidation event within the fatty acid spiral is catalyzed by the enzyme acyl-CoA dehydrogenase. We are going to be oxidizing this single bond between the alpha and beta carbon into a double bond. Like so. And while we do that, we are going to be reducing one FAD into an FADH2, money in the bank. What is the chemical logic of this step? We are extracting energy from a carbon-carbon bond. And that's going to result in this trans delta 2 enoyl CoA, highlighted in orange. And remember, the delta 2 means it's unsaturated. The number 2 means that the double bond begins on the second carbon, which is, in this case, the alpha carbon. Tugging along, the second enzyme in the fatty acid spiral is enoyl CoA hydratase. This enzyme's job is to add and activate a water molecule onto the double bond that we just formed on trans delta 2 enoyl CoA. The ensuing product is going to be the beta alcohol L-beta hydroxyacyl CoA. L-beta hydroxyacyl CoA. All right? We can hopefully begin to appreciate why the beta carbon is going to start becoming a little bit more interesting now that we've attached an alcohol to it. And what is the logic of the second step? We're not getting energy out of this molecule, but we're preparing it for a subsequent second oxidation step. Alcohols are easily oxidized, and they're also easily made from double bonds, and that's exactly what we're doing here. Step 3 is going to feature the second dehydrogenase, L-hydroxyacyl-CoA dehydrogenase. It is going to convert the beta alcohol we just generated into this beta ketone, shown over here, yielding what is known as the beta-ketoacyl-CoA intermediate. What is the chemical logic of this second oxidation event, this step 3? It's actually twofold. One, we extract more energy. And that's because by oxidizing the alcohol to a ketone, we will be reducing this time an equivalent of NAD plus into NADH, money in the bank. But additionally, we are producing a very unstable beta-ketoacyl intermediate. Why do I think the beta-ketoacyl is special? This here is the key to beta oxidation. What we have just done is generate a second, highly electron withdrawing, highly polar functional group on this other side of the alpha-beta carbon-carbon bonds. We've got a C double bond O over there, a C double bond O over here. These are going to act like electron vacuums. It's going to make these normally rather stable carbon-carbon bonds a little bit tenuous, all right? And they're going to become very electrophilic and very reactive. This is going to make the final step easy, where the enzyme thiolase comes in and breaks the fatty acid fragment between the alpha and beta carbons. This is going to release acetyl-CoA. We do this by cutting the bond right over there. And then the enzyme thiolase will stick a replacement coenzyme A and attach it to what used to be the beta carbon, giving us fatty acyl-CoA all over again, but this time shortened by 2 carbons, an acetyl-CoA unit. The logic of the fourth and final step of the spiral is to regenerate fatty acyl-CoA, while of course making acetyl-CoA that we can use for further oxidation purposes. And by now, I hope you can appreciate why we call this the fatty acid spiral, because we are going to continue this process in successive cycles until we are down to that final acetyl-CoA fragment, and then we're done. We have fully catabolized our fatty acid into acetyl-CoA units. So, beta oxidation, it's quite involved. Hopefully the individual reactions weren't too confusing, And after seeing all of that, I think it's very natural to ask, what's the bang for our buck here? What do we get for all of that effort? Let's assume moving forward that we're tallying the ATP yield for one C16 palmitate. And when we activate it, it's actually not palmitic acid, but palmitoyl-CoA. So how much energy do we get out of that? Let's distill the products of the fatty acid spiral down into three simple rules. The first rule I want you to understand is that every spiral is going to subtract one acetyl-CoA unit from the fatty acid being digested. So each spiral is going to reduce it from C16 to C14 to C12 and so on. The second rule is that one spiral will generate one equivalent of NADH and one equivalent of FADH2 because there's two oxidation steps. The third rule, in my opinion, is the most tricky. The spiral will end when the fatty acid is cleaved into two acetyl-CoAs. So what do I mean by that? The first rule is we're going down in increments of 2 carbon units, right? So looking at palmitoyl-CoA, you may think, well, we can do the fatty acid spiral 8 times because 8 times 2 is certainly 16. However, we have to think about the last step of that fatty acid spiral. We are going to have butanoyl-CoA and it's going to be oxidized over here into the beta-ketoacyl, and we are going to chop off the bond right over there. And that's going to splinter this off into 2 equivalents of acetyl-CoA. This portion and this portion. It's got 2 carbons, all right? And we're going to replace this whole portion with another coenzyme A group. So that's why we only actually get 7 turns of the fatty acid spiral for something that is C16, like palmitoyl-CoA. The last step is going to generate 2 acetyl-CoAs at once. So let's create a little table down over here of all the goodies we get out of one bout of fatty acid oxidation. This portion over here is going to represent the 7 FADH2s and NADHs we make upon doing 7 churns of the fatty acid spiral. Now we need to convert those into ATP units. And remember that one NADH is worth 2.5 ATPs, and one FADH2 is worth approximately 1.5. So we just multiply. And get these equivalents of ATP formed. We haven't considered yet, though, what happens to the 8 units of acetyl-CoA generated from 7 churns of the fatty acid spiral. Those are going to feed into the citric acid cycle. And recall that for every acetyl-CoA pumped into it, we're going to net 3 NADH molecules and 1 FADH2. And we get those specifically from isocitrate dehydrogenase, alpha-glutarate dehydrogenase, malate dehydrogenase. And succinate dehydrogenase is going to give us the FADH2. We can use the same PO ratios to tally out the 3 times 8 NADHs we've generated, along with the 1 times 8 FADH2 generated. And we can't forget succinyl-CoA synthetase, which is very peculiar as it makes 1 ATP or GTP per churn of the citric acid cycle. So that's where these 8 ATPs come from. And then you add all of these together to get a whopping total of 108 ATP molecules per palmitic acid catabolized. The math isn't hard, but it's a lot of numbers, so I highly recommend you pause, rewind, and check the math out for yourself if I went too fast. Because the overall equation for this chemical reaction is indeed complex, and I've compiled it for you up here in this corner, where 1 palmitoyl-CoA plus 23 units of oxygen, 108 inorganic phosphates, and ADPs each are going to become 1 coenzyme A, 108 ATPs, 16 carbon dioxides, and 23 water molecules. I've highlighted that for a very special reason, by the way. Fatty acid oxidation not only produces a lot of energy, but it's going to generate quite a bit of water as well. And again, that's going to be very important for hibernating animals. A little bit food for thought. If you're sleeping, you're not drinking. So by burning energy, that's actually how these sleeping, hibernating bears over here are able to stay hydrated. They won't have to disturb their rest to drink every 3 days or so. It's yet again another piece of proof that the ingenuity of biochemistry knows no bounds. When all is said and done, we can derive quite a bit of energy from fat molecules. Remember that 40% of our daily energy made comes from beta-oxidation. This chemistry isn't perfect, though. The brain, for example, among other tissues, is not programmed well to catabolize fatty acids. The brain in particular prefers glucose because glucose is quick to get, quick to burn, and neurons need energy now. Beta oxidation may be very ATP efficient, but it's slow. And slow does not cut it when you're trying to keep your brain alive. So what happens when the body runs out of glucose? We need to give the brain and the rest of the body something to run on. And it turns out the liver, always coming in clutch, has a way of exporting the acetyl-CoA it makes from, let's say, beta-oxidation to the organs that need it in a glucose deficit. But even that solution has a problem because acetyl-CoA is used in a bunch of pathways. It's very prominent in convergent metabolism. If the liver dumps it into the blood, it could get used up by any cell for almost any purpose, energy or non-energy. And that acetyl-CoA may get depleted by the time it reaches the tissues that need it the most, like the brain. So the liver needs to package acetyl-CoA in a certain way that tells the body this molecule is only to be used for food. And it does that by converting acetyl-CoA into what's known as ketone bodies. The examples of ketone bodies are acetone, acetoacetates, and D-beta hydroxybutirates. The body knows when it sees these molecules in blood that only the hungriest tissues should take it. Here I will outline the process of ketogenesis. This is known as an intrahepatic process, which means it only occurs in the liver. First, the enzyme thiolase. This is the same thiolase from step 4 of beta oxidation, by the way. It's reversible. And it will indeed reverse the last step of the fatty acid spiral. It's going to reform the last beta ketoacyl intermediate, which in this case is called acetoacetyl-CoA. I know, it's a little bit of a tongue twister. The next two enzymes, HMG-CoA synthase and lyase, will help remove the coenzyme A from this intermediate to form acetoacetate. So after we do this and we do that, we will end up with a ketone body, shown in this orange box. Check. Acetoacetate, though, I should mention, is what I would call an unideal ketone body. That's because beta keto acids tend to be prone to spontaneous decarboxylation. In the beta position, we have a ketone. Alpha, beta. See that? This CO2 group is a really good leaving group, and you can and will have that CO2 pop off, just like so, very entropically favored, and you will convert acetoacetate into acetone. The body can't use either of these products, and they end up being wasted and exhaled. They're very volatile. So acetone, it really doesn't count. The body, however, is aware of this, and it will try to divert some of the acetoacetate into beta hydroxybutyrate, shown over there. The enzyme D-beta hydroxybutyrate dehydrogenase will use an equivalent of NADH and convert it into a more stable ketone body. And that's a way that we can prevent the spontaneous, wasteful decarboxylation of acetoacetates. Ketolysis, the reverse of ketogenesis, is what we call an extrahepatic process, which means that the regeneration of those two acetyl-CoA units that we initially packaged into the ketone bodies is going to occur predominantly outside of the liver. If you look at the intermediates and the enzymes in the catabolism pathway, it's a near inversion of ketogenesis, with the exception of this central Scott-catalyzed step. The process of converting acetoacetate into acetyl-acetyl-CoA is the only unique step here. And this enzyme Scott is not made in the liver. Scott, what it's doing is the reverse of the liver's combined HMG lyase and HMG synthase steps, all in one fell swoop. So Scott is basically the equivalent of the lyase plus synthase. Steps 2 through 3. Now you may be asking yourself, well, wait a minute, for ketogenesis, why did we do this transformation with two different enzymes when ketolysis is able to do the same thing in reverse with just one enzyme? Doesn't that seem inefficient? Well, think of it this way. So far, we've learned that the liver is like the body's fairy godmother. It dishes out glucose, fatty acids, acetyl-CoA whenever the body needs it. It accumulates and releases fuel reserves. And because the liver is the only organ that lacks SCOT, this prioritizes the role of the liver as an acetyl-CoA giver rather than a taker. And that's because SCOT, if you notice, is irreversible, and all of these other steps are reversible. The combination of ketogenesis and ketolysis is known collectively as the starvation response. And it's important that I stress that this is a temporary solution to the problem at best. One of the many issues that stems from a chronic glucose deficit is that these ketone bodies are going to accumulate. The body can't metabolize them that quickly. And the funny thing about ketone bodies is that they're actually not best described as ketones, except for acetone, which is the most useless of the ketone bodies. Ketone bodies are actually organic acids, acetoacetates, D-beta hydroxybutirates. So when the liver pumps these compounds out because we've run out of glucose, what's that going to do? It's going to cause your blood pH to drop. If this goes on long enough, it can result in a condition known as ketoacidosis, and this can be a life-threatening condition. It's a particular problem for type 1 diabetics who must often monitor their urine with test strips, like shown over here, that can detect these ketone bodies accumulating in the urine and let the patient know that it's time to maybe adjust their insulin schedule. So indeed, the starvation response works short term. Our lipid reserves are an effective second resort of energy when calories are low, but don't forget that our bodies are only designed to use beta oxidation to cover 40% of our daily energy needs. If we lean into fatty acid catabolism too much, the consequences can indeed be dire.